Hi everyone. So this video is going to be short and sweet, and it's over one of the first labs that we do in microbiology, which is called the Ubiquity Lab. And so whenever we do labs in the virtual setting, you want to make sure you have the lab worksheet. So for Ubiquity, it looks like this. It provides all the background information and any procedural details that you need to know for the lab, because even though we're not face-to-face, you still want to make sure you understand the purpose of the lab and what we did, right? Because that goes hand in hand with the purpose, and then understand why we got the results that we got, okay? So you don't necessarily have to memorize every step of the procedure, but you want to understand what we did so you can understand the objective of the lab and, of course, understand the results. So to start this lab, I want to show you this photo, which went viral a few years ago. And somebody brought a Petri dish to some restaurant or convenience store somewhere, and they placed it under one of those hand dryers, the ones that blow hot air at you. And then they incubated the plates, they let organisms grow, and this is what they got. And it went viral, and people panicked about, oh my gosh, these are so unsanitary, we can't use them. And so one of the big objectives for me, at least for this class, is to teach you to kind of push pause and think critically and scientifically about what you're saying. Because it is very natural to look at this picture and go, OMG, what is this? What am I putting my hands under? But we know that microorganisms are everywhere, right? They're in the air around us. They're on all the surfaces we touch. They're in water and soil, right? They're everywhere. That's what ubiquitous means, everywhere. Okay, and so we know that microbes like bacteria and fungus and viruses and things are all over the place. Okay, so really it shouldn't be surprising that we get a lot of unattractive organisms growing on a plate when you're blasting hot air at it, right? Because that hot air is moving any organisms in the vicinity down and onto this plate. And of course, anything that was living on the surface inside the hand dryer. Now, just because something grows ugly and scary doesn't mean it actually is dangerous to our well-being, right? We do go into these restrooms and we use these hand dryers all the time, and we don't typically get sick, right? So yes, it's not a pretty picture, but this doesn't mean you should never use a hand dryer, okay? All right, so let's quickly walk through the background of this lab. So I've already told you that microbes are ubiquitous. They are everywhere, okay? Remember in one of the content videos, I talked about our normal microbiota, which is also called our normal flora, but I tend to use the term microbiota. And those are the organisms that live on our skin or in our bodies, and they're helpful to us, okay? And we're gonna grow some of them, and they're not the prettiest looking species either. But in the laboratory setting, this is where we would be doing our micro labs, there are four main forms of contamination. And if you want to be following along, you can look at our worksheet here, okay? So the four biggest sources of contamination in the laboratory, but also these are common sources of contamination just in general, are the skin, right? We have lots of microbes that live on our skin. And so as you touch things, you can potentially contaminate your workspace, yourself, your cultures. So that's why we always wear gloves and we're very careful. Another big one are respiratory droplets. So even though you can't always see it, every time you're talking, you're sneezing or coughing, you're releasing aerosols out into the environment. And so a lot of times when I do inoculations, which is transferring, bacteria from one media to another, I don't talk because I don't want the contaminants in my mouth to get into my culture. Because especially when I was in grad school, that could ruin your entire experiment. Okay, so a lot of times we don't talk, and to be honest, I had some critical experiments where I wouldn't even breathe if I had a test tube open or a petri dish with an agar plate open, because I did not want to risk the integrity of my experiments. You just kind of hold your breath a little bit. Another form of contamination is the air, because again, there are microbes everywhere. As you saw from that hand dryer photo, a lot of those came from the air. And so when we're in lab working, anytime you take a lid off of a test tube, you move quickly, safely, but quickly, because you're trying to minimize the amount of contaminants from the air that could potentially get into your culture or on your inoculating loop, which is what you use to transfer bacteria from one place to another. So you just have to be very, very careful. And then the last one are inanimate objects, right? If I were to sneeze and then open the classroom door, anybody else who touched that handle could potentially get my germs. Same thing with, right, with anything in the lab. So those are our 4 big sources of contamination. Okay, so for this lab, we want to show you that microbes are everywhere. And so what we would normally do is get a petri dish that has nutrients and agar in it. We call that solid media. So when there's solid media in a petri dish, we call it a TSA, trypticase soy agar, or normally we'll just say an agar plate. So we would take several agar plates and we would label them. You always label your media, but more on that in a future video. And notice there's a plate where it's to look at what's in the skin. So you just take, I would have taken the lid off this plate and just set it on my desk as I lectured to y'all for the class period. Then you put a lid on it and set it off to the side. Then we'd take a second plate and divide it in half. And you would touch the agar on the before side with a dirty finger. And then you would go and wash your hands and then take your clean finger and rub it on the other side of the plate. The third plate is a kiss. Now, agar plates, when they come out of the autoclave, the media is 100% sterile. It is not a risk. People always look at me funny when I'm like, kiss your plate. And they're like, oh, no, thanks. But it's safe. It's probably safer than anything that you could put your lips to. And then you pick an inanimate object for the fourth plate, you'd swab it and swab it onto the agar plate. So then you'd put all of those plates, you actually invert them. Well, actually, that's not necessarily true in this lab. You'd take those plates and you'd put them in the incubator, give it a couple of days, and then we'd look at it, okay? And so we're gonna come back to these questions in a minute. Let's go ahead and look at the lab results first, and then we'll use some of the remaining text to help us explain what we see. Okay, so moving on, here's the Ubiquity Lab results. So this is what grew on the plate that had been exposed to the air. Now, what I want you to notice, kind of a quick plug for future labs, let me get the little pen here. Do we agree that these 4 growths, right, look identical? This one on the left here is a little smaller, but that's just because it ran out of space, right? It's wedged between one organism and the edge of the dish. So when you see growth that looks identical, that means they're from the same species of organism, okay? But notice we have this organism right there, right? It looks like it's pretty fuzzy as well. So these are all types of fungus. But this one didn't grow near as big, and it's got kind of a darker color in the middle. So because this growth over here, this little small one, does not look like the other 4, we would say this is a different species. Okay, so all of these guys, I'm just going to call them species A. I haven't tested them. I don't know what they are. But this would be a different species, because as you're going to see later in the class, different organisms grow differently on an agar plate. For instance, this goes on your handout. Staphylococcus aureus is an organism that is commonly found on our hands, and it grows as a gold color on an agar plate. And you're going to see it here in just a second. But Staphylococcus aureus is one of the only organisms that grows as a gold color on an agar plate. So that one, you can look at it and go, oh, that's gold. I know what that is. Okay. But things like fungus, At least personally, I don't know the different types of fungal species based on their growth. I'm more of a bacteria person. All right, so I mean, the air isn't too bad, right? At least when there's not this forceful ejection of hot air. All right, so let's look at the lip and nose imprint. So down here on the bottom, let me see, I think there we go. This was the kiss on the agar plate. And so this is all the bacteria that is typically on your lips. Okay, isn't that fun? And then up here, this is a nose imprint. So just put the tip of your nose onto the plate, and that's what you're going to get. So these are different species of organisms that are just commonly found on the tip of your nose. All right, so people tend to think that there's nothing really on them, right? You can't see the microbes. You don't tend to think about it, at least. But this is just to give you a nice illustration of what is on and in your body. All right, so the next one are inanimate objects. So we swabbed the keyboard and look at some of the fungal and bacterial species that grew. Okay, so this is just looking at it from the bottom of the agar plate, and this is looking at it from a top view. Okay, and so I mean, think about it, right? I'm sitting here making this video and my keyboard is in front of me, and even though I'm not typing on it, I'm talking, right? So things from my mouth, those aerosol droplets can be getting onto my keyboard. And then of course, as I touch my keyboard to type, right? I don't go wash my hands before I touch my keyboard every time. So there's lots of different organisms present on the keyboard. Now check out this one, the bottom of a shoe. Look at all these different species found on the bottom of a shoe. Beautiful, huh? Okay, let's go and wrap this one up. So this was the before washing hands is on the left. And notice those gold, they almost look like perfect circles, right? So a growth on a plate, we call it a colony. Okay, a colony is a mass of cells on a plate. So let me see, is the pin still on? So this is a colony, this is a colony, this is a colony, this. Okay, so these little individual growths are called colonies. And so Staphylococcus aureus are these gold colored colonies. Okay, so then these white colored colonies, some of the ones that I was checking off, that would be a different species of bacteria. Okay, so and again, it's not surprising that Staphylococcus aureus and these other bacterial species were present on the hands before washing them, right? Because they just naturally live there. And then by doing a quick wash of your hands, look at how much we reduced the amount of bacteria on our hands, right? It's pretty snazzy. Okay, so let's kind of wrap this up. So you can answer these questions on your own. If you have any questions, y'all, please ask me, okay? I'll probably actually give you the answers here at the end. So I'm gonna pause a little bit, push pause on the video, try and answer them yourself, right? Because this is going to be a really good study guide for the lab exam, okay? You need to know what's on these handouts. You need to understand the results, but these questions are also really good for you to know. Let's see, common species of bacteria on your skin, okay? They were the ones we were just talking about as we were going through and looking at the before and after hand washing plate. So that's going to be Staphylococcus aureus, and they are a gold color. Okay, we've already done 1 and 2. Ubiquitous means they're everywhere, and the 4 major sources of contamination were in that top paragraph. All right, so down here, this is just a nice background on the human normal microbiota or normal flora. I talked about them in the 3rd content video for Introduction to Microbiology and the different types of relationships. So listen to that video, read this paragraph, it's really helpful, okay? And make sure you can distinguish between the types of relationships for the host and the microbe, right? Our body, when we're talking about like getting sick, the human is the host, okay? And the microbe is the microbe. Now, but remember, not all human microbe interactions are negative, right? Our normal microbiota is a very mutualistic, a beneficial relationship. We help the normal microbiota and they help us, okay? Then down here at the bottom are some common normal microbiota species and where they tend to inhabit. So you'll notice In this case, there's a lot of bacteria. Really, the only fungus that I see just skimming it is down here at the bottom that I talked about in the in the content video when I talk about opportunistic pathogens. I talk about how yeast can be a problem in the vaginal region. So go check out that content video. It's a very important idea that we will be coming back to a lot. But just look at the diverse species that exist in different parts of our body, okay? All right, y'all, let me know if you have any questions.